starting. So it is my pleasure to introduce Otis today. So Otis has done his undergraduate degree in Plymouth and then graduated and moved into the Deep Sea Crew, which is an amazing research team led by Professor Kerry Howell, and that's where he kind of delved deeper into the marine world. He's doing some really interesting research on connectivity in the deep sea, and this is absolutely foundational. So we have very little comprehension of how well different areas of our deep sea are connected. So the work that Otis has been doing is actually, really pushing the boundaries and opening up windows into how this mysterious, dark, very far from our world environment that is the largest habitat on the planet exists, and how those different areas are connected. The research that's coming out is amazing, so I'm absolutely thrilled to have Otis, who's come a very long way, by the way, Okinawa Institute of Science and Technology, which, looking at it, isn't a terrible place to be based for a while. So yeah, we're thrilled that you're here with us, and I'm very much looking forward to this. So thank you very much.
Thank you for that excellent introduction. I should mention before I start about the Institute a little bit, because the funding to get me all the way from Japan here today came from the Institute. This is what they call an educational visit, which PhD students can partake in, which is funded. And but it does require me to talk about the Institute a little bit. But the fact, the fact that they funded a PhD student to come all the way to England, and that it looks like that, I feel like is advertisement enough, so I won't spend any more time on it. So broadly, I want to talk to you guys about hydrothermal vent connectivity. Mostly I want to talk about connectivity, but I work with hydrothermal vents. So yes. So a quick background to the talk. First, I'll start with a background, a bit about myself, the ecosystem in question, and then the first section of the research is about the biodiversity at hydrothermal vents. The second is about the dispersal among hydrothermal vents, and in the final section of the talk, I combine that into the idea of metacommunity dynamics at hydrothermal vents. So a little bit more building on from the background that Michelle already gave you. I did my undergraduate at the University of Plymouth and spent my time with the Deep Sea Crew making predictive models of many deep sea ecosystems, including this Lophelia or Desmophyllum pertusa, this cold water coral reef, and where it's distributed around the UK based on the environment that we find it in. And I created a predictive model that shows in red here where you're likely to find it with some high probability. Actually, I use this slide as a demonstration of my research highlight. So this position, this research assistantship, culminated in an expedition on the James Cook, incidentally where I met Michelle, and we were looking for these coral reefs. We were looking to sample them for genetic data, and this is what the sampling looked like. We would send down an ROV. Oh, sorry, I'm supposed to use the laser pointer. We'd send down an ROV, and we would do some transects in yellow here. But this particular transect map is particularly important to me, because this is where I had my research highlights, where we sent down the ROV around this yellow area, and we found this soft sediment, sandy bottom, and we were really hoping to collect some corals from this area, but they don't live here at all. So me and a colleague ran upstairs, this was the middle of the night, and we checked our predictive models, like you see here, and found that if, rather than abandoning the dive after doing the transects and going to the next site, if we just do some transit with the ROV all along this black line up to here, where the slope is a bit steeper, our predictive models agreed that we would find some coral reefs there. And then, because I was on the night shift, I went to bed, and after 8 hours of transiting in the open water by the ROV, it landed on this coral mound. And waking up in the morning at the time it was supposed to get there, and my predictive model said it would be there, it's still a research highlight of mine. So I wanted to share that. Not particularly sure if it's important to the story at all, but I just wanted to share that. Actually, no, it is important, because this was the Deep Links research project, which was my first experience working on connectivity research. And then after that, actually right after that, I went to Okinawa in Japan. This is a small island in the very south of Japan, closer to Taiwan than Tokyo. Subtropical climate, jungle, beautiful, beautiful island. And also surrounded by hydrothermal vents, these little red-orange volcanoes here. So a quick mention about what hydrothermal vents are. They are, well, hydrothermal vents basically means where hot water comes out. So that's what they are. They're where seawater is superheated in the Earth's crust, and that is expelled, vented through the earth, bringing with it chemical-rich water. And these ecosystems are important for connectivity and important in general and scientifically interesting because they're chemosynthetic ecosystems. Their primary source of energy is from the chemicals venting out of the Earth's crust, not from sunlight like most ecosystems on the Earth. So they're very well self-contained, they're very interesting, and they may be the origin of life on Earth and also on other celestial bodies in our solar system. It's hypothesized that hydrothermal vents could support life independently from sunlight on moons orbiting Saturn and Jupiter. So this is the slide I like to show to demonstrate to people why hydrothermal vents are interesting beyond the research that I did, which has nothing to do with any of that. Hydrothermal vents were discovered about 70 years ago, and these geologists went there to go to these areas they'd hypothesized exist at tectonic boundaries. They went there and they found that they did exist, but what they didn't expect to find was that life also existed and thrived there. Actually, the geologists on board were so ill-prepared for biological specimens that they actually used their own private stock of vodka to preserve the first biological specimens from hydrothermal vents, which is very resourceful of them. But what we found since then is not only does life exist, but it is abundant at hydrothermal vents. Oh, the videos are terrible by Zoom. Okay, well, we'll move on. That's a lot of shrimp, if you can't see that on Zoom. And not only is it abundant, but very diverse. Different hydrothermal vents have very different species living there. So also, why do I study hydrothermal vents? Well, when I started my PhD in Okinawa in 2017, was also the time that the world's first large-scale mining of any hydrothermal vent ever happened. Happened right in Okinawa in September 2017, with plans to commercialize mining, so that means make this large-scale mining continuous in 2020. Some things happened in 2020, I don't know if any of you noticed, but that got pushed back a bit, and full-scale commercial mining hasn't begun yet, but from their tests in 2017, they're ready. So this got me wondering, what does this mean for the species that live at these hydrothermal vents? Are they able to move? Sorry, laser pointer. Are they able to move and colonize other nearby hydrothermal vents? And what does this mean for Japan's very serious commitment to sustainable development goals? Really wish the videos were a bit, but you can see the guy, the pineapple pen, advertising sustainable development in Japan. Taking it very seriously. This topic is really great topic to communicate to people in Japan about the local ecosystems and travel to schools. And I got some funding to make physical prints of this Treasures of the Deep book, which is wonderful, which is in Japanese also, and we're distributing these across Okinawa. Hopefully people become more aware of these ecosystems that they have that are in danger. So onto my research. What I'm going to talk to you today is effectively my PhD thesis. In my thesis proposal at this institute, you design and propose your own research thesis, which was really a wonderful experience. In the first chapter, I said I would describe the biodiversity at hydrothermal vents. In the second chapter, I said I would simulate dispersal among these hydrothermal vents. And then in the third chapter, I would sort of model or look at the relationship somehow. I actually haven't figured it out at that point, but it seemed convincing enough to get me through it. So to characterize the biodiversity, I originally was going to use openly available data, but then got collaborating with local taxonomists who told me that the publicly available data was garbage. So they provided their own. Chong Chen provided a new data set for the Okinawa region, Hiromi Watanabe for the Izubonin, and Verena Tunnicliffe, who was my examiner for my proposal, she jumped on board and gave me the missing piece of the Mariana region here, which is outside of Japan and in the Americas, actually. So I just brought them together. I didn't actually do any of the taxonomy. I'm really heavily reliant on them for this first chapter. So massive shout out to them and all the taxonomists. So this data looks like a site by species matrix. I only had presence and absence because hydrothermal vents are pretty hard to get to and hard to confirm the actual abundance. Actually, this isn't presence absence. This is presence and pseudo absence. It's a 1 if it's present. It's a 0 if we don't know if it's present or not, because very few of these hydrothermal vents have been comprehensively surveyed. So you start to get the idea of the quality of diversity data I have here. So I made the most of it, I promise. I transformed that into a pairwise similarity coefficient. So with 1 being all the species at the pair of vents are exactly the same, 0 being they have no shared species. And I went a step further and then formatted this as what I call a species assemblage network, where the pairwise similarity is a weighted edge, this indicated by the color of these lines, a weighted edge that connects the nodes of a network, the nodes representing distinct hydrothermal vent sites. I also created a bipartite network, which is a bit different, and I'll explain why it's important. But it's effectively an unweighted network where each species is connected to each site at which it is present. So, sorry, pointer. This copepod here, I made this little cardboard thing, this little paper model myself, trying to impress you, is present at this hydrothermal vent and this hydrothermal vent. So that's what the lines in the network mean. So this is the results of my first chapter, and I forgot to plug it, but the paper is ready and it's coming out in Ecology and Evolution. This is the species assemblage network, where the lines indicate shared species similarity, the darker lines mean stronger similarity. And the colors are indicative of these clusters, how the species similarity, how this network clusters based on the number of shared connections. Basically, by minimizing the connections between clusters, maximizing the connections within clusters, and iterating that loads of times, you end up with this sort of clustering where you have, which overlaps quite well with the tectonic basins there. You have the purple, which are mostly which are all in the Okinawa Trough, the green, which are in the Izu-Bonin Arc and the Mariana Arc, and then these light blue ones here that are in the Mariana Trough. We also have some outliers. But yeah, this is effectively why I made it. The reason I made a network out of this shared species is because it allows you to not only cluster them, but also to look at certain hydrothermal vents, sorry, laser pointer, that are particularly important in connecting distinct clusters also. So not just looking at the global clustering of these vents, but also the individual role each hydrothermal vent plays. And the bipartite network, I use that for the cluster to look at the role species played in this network. So here again, here the smaller nodes are indicative of species that are just numbered, and the larger nodes are clusters of hydrothermal vents. So the purple one is all the hydrothermal vents of the Okinawa Trough, for example. And then if you look at this gives you an idea of certain species that may play a particularly important role in the region, like this snail, which is present at 3 distinct clusters. So these clusters may be geographically separated, they may be environmentally separated. Either way, this species is able to inhabit and exist at all of them. And an interesting question that leads on from this is what makes this species so special? Is its response to environmental variables very plastic, or has it got a really great little dispersing larvae that is able to reach all of them? We don't know, but it starts to give you some indication of these patterns. The main reason I used this bipartite network was to calculate the module degree. So this is the proportion of links that are within the module, the proportion of shared species that are within the module, indicated by this line, and the proportion of links that are between modules, which gives a rough indication and allows me to plot in 2 dimensions the relative importance of each hydrothermal vent in shared species connectivity in the region. So for example, these purple ones up here, Sakai and the North Knoll Ihea Ridge, they're very high on the y-axis, which indicates that they are very central to all the shared species that are indicative of Okinawa Trough. All those species that are commonly found in Okinawa Trough are almost always found in these 2 vents. And so it could be a hub for those species in the Okinawa Trough. And on the x-axis, we have those hydrothermal vents that have intermediary species that are found across different regions, like this Myoin Knoll here. And why this is important in this first paper and for conservation is because these hydrothermal vents in the central Okinawa Trough are the ones that are targeted for mining. So these ones that are targeted for mining are potential hubs of the species that are indicative to the region. Anyway, I said I would talk about connectivity, but at the moment I've just talked about shared species, and connectivity is a complex process, and it's not all, and the presence of shared species doesn't always indicate contemporary connectivity. Contemporary connectivity requires the dispersal of larvae between sites and then the post-larval settlement and survival at the next site. So how am I able to quantify this? Well, first let's start with the dispersal. So I used oceanographic data. This is terrible video again. This is the surface temperature of the region, and it somewhat shows the primary current system, which is the Kuroshio that sort of goes up this way. You can see the red dot moving a little bit. And so I take the environmental data, the surface currents, and then I use a set of methods called ocean parcels, where I drop a particle, indicated by this block, in the grid of ocean velocities, and the values of these velocities dictate where that particle goes in the ocean. And you can do it in 2 dimensions and even 3 dimensions. Parcel standing for probably a really computationally efficient Lagrangian simulation. So bear with me on this video, because this one's sort of important. So effectively what you do is you drop the particles here at this hydrothermal vent site and see where they go over time, based purely on the currents that are in the region that have been measured and hindcast over many years. This is a simulation that is frozen. This is a simulation that shows how particles would move from one hydrothermal vent at 500 meters, just purely based on the ocean velocity. And this plot shows their distribution at the end of a certain period of time, maybe 80 or so days. But how do you turn such a measure of Lagrangian, how do you turn Lagrangian particle tracking, just seeing where particles move through the ocean, into dispersal, into a dispersal simulation? Well, you need the behavior of the particles. You need to add the behavior of the particles to make them act like larvae from the hydrothermal vents. That behavior includes the planktonic larval duration, how long these individuals survive in the water column, and the depth at which they disperse. Two of the most important behavioral parameters to incorporate into this simulation to turn it into what we call a dispersal simulation. So an example, I did this for many different species and it's ongoing, but for one example, I was interested in this particular species in a group of species of shrimp, albinocarid shrimp that live at hydrothermal vents. So I created a model where we simulated the dispersal of these shrimp, but with this unknown larval phase in the middle. So this species reproduces, and its first larval stage is lepidotrophic. It just survives off its own energy stores and disperses at we assume, based on experimental observations, disperses at the depth at which it is released. But then it has this unknown stage, which there is evidence that it's planktotrophic, and also evidence that it may go closer to the surface. So we simulated this. And in this simulation, we have the larvae leaving the hydrothermal vent. And then at roughly 70 days, which we've found from experiments, it heads up in the water column and disperses during its planktotrophic stage. But the depth at which it disperses, we're unsure. So using these simulations, we can try lots of different depth hypotheses and see where that takes them. And then we assume that that the duration of its larval duration, results in this observed juvenile settled stage. The reason I'm incorporating these pictures is because it makes mention of the cephalothorac length, the CL, which is what I use to try to predict the planktonic larval duration. So here I'm showing that we try lots of different depths, but the next stage we need to try and guess the planktonic larval duration of this species where it has unknown stages that we've never seen before. So I use this model by O'Connor et al., who took data from 69 different species and their different planktonic larval duration at different depths in experimental studies. And I extrapolated that information. I parameterized that model they made into my dispersal simulation. And here I have the results of how long these shrimp larvae are likely to survive in the water column. So on the x-axis, we have the age in days. And on the y-axis, we have a maturity value, which is based on their relationship with temperature as calculated by O'Connor. And we can see that during the first 70 days, roughly, their maturity is only slowly increasing because they're dispersing at depth. And then after that, I send the particles up into the higher water column where the temperatures are much higher and the maturity starts to skyrocket. And we can see that from that, they reach their like terminal maturity threshold indicated by evidence of their cephalothorac length. They reach that planktonic larval duration, that terminal phase, somewhere between 120 and maybe 220 days. So it can be very variable based on the ocean currents in the region. Yeah, like I said, it's based on observations of how large they are at the time at which they settle. So to ground truth what we did, first I take one of these, this is the output of such a simulation. This is the probability density function, the density of particles, or sorry, the density of larvae now, where they reach their planktonic larval duration, if they were released from this single hydrothermal vent site. The reason I chose this one is because in a few years before, in an experiment I like to call Finding Nemo, Nemo floats were deployed at this hydrothermal vent to move with ocean currents at 1,000 meters specifically. So the same depth at which I run the simulation, they would disperse at 1,000 meters. They would then pop up to send off their location, then go back down to 1,000 meters every few days. And so from that, we can find, we track their distribution through that time and see that there's roughly an overlap. They're sort of plotted in different perspectives, but you can see that it sort of roughly agrees with the distribution of the particles that I released from this hydrothermal vent. So this is our way of ground truthing it. A lot of time and money was spent in sending off these Nemo floats, so I'm happy to reuse that data for ground truthing. So the next stage is turning these dispersal simulations into something that I can analyze and compare to my empirical observations from the first chapter. This is done by looking at the probability density function, again, from, let's say, one vent, from vent A, and looking at the density at vent B, and using that density as the probability of dispersal between vent A and vent B. And then I reiterate the simulation, releasing them from every vent site and seeing at which vent sites they end up at, and I have another network. This here, the lines indicate the probability of dispersal between pairs of hydrothermal vents. And you may see some similarities to the network I showed you before, which is the pairwise similarity of species between hydrothermal vents. So qualitatively, you can see that there's some agreement in the dispersal probability and the shared species, which is what we expected. Also, just a quick note, just minor changes to the dispersal simulation can result in quite large changes or visible changes to the dispersal probability between them, sort of flicking between them in the left slide here. So my simulations show larval transport, and I assume that that larval transport results in settlement and therefore equates to dispersal simulation. But so that's what this is showing here, is showing a dispersal simulation, but dispersal is not the same as connectivity. Connectivity requires successful settlement and reproduction at the target site. So how do I take these estimates of dispersal and try and calculate or try and quantify connectivity without any genetics data, basically, to ground truth and understand and really quantify connectivity between vents? So I turn to metacommunity theory, and don't worry about the equation, let's just call it metacommunity dynamics. Metacommunity dynamics, the interaction between spatially isolated communities is dependent on these three factors. It's dependent on abiotic responses, these local environmental variables, density-dependent biotic interactions, the interactions between the species at each site, and dispersal. And I'm just focusing on dispersal at this point and just make some assumptions for these other two. So to do this, I employed a meta-community simulation that was made by a colleague of mine, actually a fellow graduate from the university, the Okinawa Institute of Science and Technology. I won't go to too many details into how the simulation works. And for those of you going to BES in a few days, that's where you'll hear about that. But effectively, we release 100 different simulated species indicated by this model, also I made. And so effectively what we do is we release these 100 species and see where they go in the dispersal network that I created. They're able to move between these habitat patches based on the dispersal probability between them. So this is how I incorporate my dispersal networks into metacommunity dynamics. So we run this for with lots of different species, 100 or so, and then we look at, then we transform that data of where they ended up into the same pairwise shared species between each hydrothermal vent. And again, I turn that into some networks, and these networks show the similarity between each hydrothermal vent site in the dispersal network based on where these simulated species ended up. The reason I do this again in the simulation is because I'm able to iterate the importance, the relative importance of dispersal strength on the x-axis and the relative importance of local selection strength. This is the environmental filters. I'm able to tweak those in the simulation and then see what that results in. And that results in these networks with one of them in particular looking quite qualitatively similar to the one from my first chapter. Here, the nodes are colored by their tectonic region, the Okinawa Trough in pink, Mariana Arc in green. And as we found from this empirical metacommunity network on the right, the Okinawa Trough is very separate from the rest. It's connected via the Izu-Bonin Arc and Mariana Arc, and the Mariana Trough is quite separate. So qualitatively, we can recreate this in this simulation using my dispersal network and this hypothesized interaction between dispersal and local environmental filtering. The reason this is important is because as we iterate this, we can see the meta-community paradigm that's really driving the community composition regionally for hydrothermal vents. So two non-mutually exclusive paradigms are species sorting and mass effect. Species sorting says that species will end up wherever their environment is best in the meta-community, while mass effect says that this can be overridden if there's enough input of a species into an environment. Effectively, species can exist in environments that are suboptimal as long as there's a strong enough dispersal input from an optimal site or habitat patch. So by changing the role of dispersal and local environmental selection, we can change how this simulated network is driven by different meta-community paradigms, and then see where our dispersal network ends up on that. So we can see that the one that looks most qualitatively the same to the empirical network falls somewhere towards regional exclusion in the mass effect. This means that the strength, they have relatively weaker dispersal strength that would, well, let me rephrase that. Stronger dispersal strength and stronger dispersal probability among the hydrothermal vents would actually lead to better local coexistence and higher beta diversity across the region. But these hydrothermal vents are more strongly impacted by local environmental filtering and weak dispersal, relatively weak dispersal that results in some species just being excluded from the community entirely because they can't reach their optimal environments. Another fun aspect of recreating this meta community in a simulation is that we can then incorporate anthropogenic disturbances. On the right, we have the surface temperatures again, and we can use forecast predictions of global climate change and global surface, ocean surface temperature, run this dispersal simulation with higher temperature, which would affect the planktonic larval duration and potentially reduce dispersal, then use that dispersal network to recreate the simulation and see how it looks different now under hypothesized climate change. Also, we can just remove one of these hydrothermal vents from the dispersal network and effectively recreate a worst case scenario of hydrothermal vent mining and then see how that affects the meta community, how that affects the connectivity across the region, how that affects the diversity across the region. Oh, yeah, okay. So in conclusion, regional scale impacts from mining depend on the target vent's role in connectivity. As I showed in the first chapter, some hydrothermal vents have a particularly strong and important role in sharing species across the region. So the impact of hydrothermal vent mining will regionally will really depend on which hydrothermal vents are chosen. And I'm hoping that this information, this publication, can inform that choice, because maybe in international waters, hydrothermal vent mining is a maybe, but in Japan, it's gonna happen. They have control of their own region, they're going to do what they need to do, and Japan is famously resource limited. So they're going to extract these minerals from hydrothermal vents. And potentially this information on the role, the relative role of each hydrothermal vent could inform the destruction of a less important vent. But you can see that's a bit of a reach also. My second conclusion is connectivity is complex, but certain aspects of it, particularly dispersal, probably should be simulated. Other aspects also like this metacommunity dynamics can be simulated, but there are a lot of assumptions involved. So it's an ongoing process. And finally, simulations like these, I think, are particularly important tools for remote ecosystems, those in the deep sea, those in hydrothermal vents, but generally most ecosystems are quite remote in the marine environment anyway. So we sort of require these simulations to predict future impacts because we're not going to get the necessary data anytime soon. So I want to say thank you to everyone who supported me. Well, there's more people than this that supported me, particularly to Dr. Suzuki and her meta-community model. Also, I wanted to plug this portal that me and some collaborators are working on called See the Connectivity, where we take our connectivity data, whether that's in the form of dispersal probability network, shared species probability, shared species similarity, or genetic connectivity, for example, and plot that as a network in space so people can see the connectivity networks around their home and in other areas of the world. I think that networks are a really intuitive way to present your data, and we're hoping to make this platform public and available to everyone and fill it with all sorts of types of connectivity data so that members of the public can understand it intuitively, but also that policymakers can see it and actually they're able to simulate disturbance events by removing the nodes, sorry, laser pointer, by removing the nodes. Once you remove the nodes, the certain aspects of the network will change. The connectivity probability will change, but also the size of the nodes changed. If you noticed, this side is this size of each node is based on some structural statistics, which may indicate the conservation value. So you can infer how conservation values of each site will change as you remove each node. So it's interactive and people can understand it in an intuitive way. Also, I've been adding this recently. I'm currently looking for a postdoc. So if you know someone that wants to facilitate, like add this to their favorite ecosystem, they want to do dispersal or connectivity research, but they just don't have the time, they could always just hire me. Yeah, so that's all.